I see it now. Okay.
You're good.
Nader, do
Yes.
you want to put your stuff on?
Yeah, can I start sharing?
Yeah, start sharing.
Okay.
And then I'll say a few words, about our webinar series, and then Maria's going to introduce you, Nader. So
Okay, no problem.
let's just see. Okay, awesome. All right, are you okay to start?
Yeah, I'm good. Can you see the slides?
I can see your slides. Yep. Okay, I'm going to hit
Okay,
start webinar.
perfect.
Give us
Got it.
about a second and then I'll start. Well, good morning, everybody. Good afternoon and good evening, whatever part of the world you're in right now. We are excited to have another awesome webinar today as part of our tumor section series. Um, I'd like to thank the AANS again for their help in assisting with this webinar. I'm also excited to hear this talk from a dear colleague and friend of mine, Dr. Sanai. And I'd like to thank Dr. Maria Noor Al-Huda, who also has been helping me with these webinars. And I'd like her to introduce Dr. Sanai.
Thank you very much, sir. Thank you very much for your very kind words. It's an honor to be part of this amazing webinar series and to promote more education around the world. It's a matter of real pleasure for me to introduce Professor Nader Sinai. I'm a huge fan of his work already, and I know that everyone around the globe is a huge fan and consider him a huge inspiration. He doesn't need any introduction because, on the first day when we join our residency, we are being introduced by the Nader with the Sanai, the Burger Sanai classification system. So every one of you know you so much, so very well, and consider you a huge inspiration. But for the sake of formality, I'm pleased to introduce Professor Nader Sanai, who is the current director of the Ivy Brain Tumor Center and the chief of neurosurgery at the very prestigious Barrows Neurological Institute. He's the Francis and Diana Chair in Neurosurgical Oncology at Barrows Neurological Institute, and not only he's a very competent neurosurgeon, but he's an exemplary researcher and an innovator. And all the time we are, coming across all the wonderful studies that he does. And it's a real honor to have him today to deliver a wonderful lecture and insightful talk on the neurosurgeon's role in clinical trials and neoadjuvant treatment and tissue sampling. So thank you very much and welcome to the webinar. It's an honor to have you here today with us. The floor is all yours.
Thank you, Maria, for the kind introduction. Thank you, Costas, for organizing this and leading this section. Obviously, an honor to be part of this seminar series, which I've caught a few of already and seems to be really compelling and necessary for our field. These are the disclosures, none of which will bear any impact on the content of this presentation. What I hope to do is either introduce or reintroduce our colleagues in neurosurgery as well as our trainees to the role that I think many of us feel neurosurgeons should increasingly play in drug development for brain tumors. As Maria mentioned, many of us are very proficient and facile with the data around surgical neuro-oncology, and so we understand the limits of it and where there's a huge unmet need in the role for adjuvant therapy and its efficacy in our brain tumor patients. If we step back and think about how adjuvant therapies come to be, many of our concepts are derived from the medical oncology community and the historical precedent that's been set for other cancers, where oftentimes we have this bench to bedside conceptualization with discovery-based science occurring in the preclinical space and then being ushered through phases 1, 2, 3 clinical trials towards an efficacy signal. One of the challenges in trying to understand where we have fallen short in developing new therapies occurs in how we ultimately, in reality, usher these new potential agents to late phase trials. So I think it's instructive to look back into the recent history at the phase 3 trials, which really represent our greatest hopes for neoadjuvant therapies, for example, for glioblastoma, and how they've arrived at that stage. This is an analysis that was recently published in the literature demonstrating that of the 20 phase 3 trials completed over a 15-year period in recent history, 3 of them, of course, we're familiar with as being successful, These are the drugs, the studies that gave us temozolomide, gave us tumor treating fields, and gave us sort of abbreviated regimens for older patients. But amongst the entire population of these phase 3s, only a minority of them appropriately utilized phase 2 data to support that decision-making cycle to enter phase 3. And in fact, of the 20 phase 3 studies completed in this range, 6 of them had no phase 2 study. 3 of them had a phase 2 study in support, but one that was probably suboptimal in its study design. A couple of them really overestimated at a statistical level the expected benefit of the agent. And a couple of them also didn't meet the predetermined efficacy endpoint of the phase 2, but nevertheless went on to a phase 3. So at its outset, what this suggests is that there are some fundamental problems in how we're evolving a potential experimental therapy from an early to mid-phase range into a late phase study. If we look at it a little deeper with a more specific lens on what we know to be a real fundamental root cause of drug failure, which is tumor pharmacokinetics and tumor pharmacodynamics, there's only one study in a similar range. These are 15 phase 3 trials that were initiated just over the last decade, beginning in 2014 and ending in 2019. Only one of these 15 actually had PK/PD data from human tumor tissue. These are drugs that span across different drug classes, and we're seeing this increasingly within neuro-oncology. But really, when we look at the basis for example, the conclusion that these 15 assets are brain penetrant, which would largely be a prerequisite for any agent having efficacy in the brain, with some exceptions. The majority of them had no human tumor PK data. The minority of them used tumor PK data from gadolinium enhancing tumor, which we know is traditionally an overrepresentation of drug CNS penetrance. And then a couple more used imaging surrogates, which is largely a difficult correlate to draw to human tissue performance. And one of them used total drug in CSF. In terms of the pharmacodynamic effects of these agents that did go on to phase 3 and are currently in investigation in many cases, only one of them had a PD analysis, and that PD analysis, ironically, was negative. And nevertheless, the drug was then advanced to a phase 3 study. If we take a step back from this and we ask ourselves, well, most of these agents had some compelling preclinical data set that drove a narrative that this drug should be promising and entered clinical study to begin with, we have to remind ourselves that in fact, across every drug class that we've tested, there's never been an example of an animal model data set that successfully predicted the clinical results of a phase 3 clinical trial. And we can look at this across preclinical data that supported targeted inhibitors, antibody drug conjugates, peptide vaccines, and a host of other precision oncology approaches. And time after time, what we see is that the preclinical data largely does not mimic what we see in humans, and for reasons that I think many of us understand very well. One of the primary tripwires for drug development in brain tumors, in neuro-oncology, and frankly in CNS human disease is CNS penetrance. And so the concept that all of us have to be aware of, neurosurgeons and beyond, is this concept of the free drug hypothesis, which is effectively this assertion that has been borne out in the data largely, that it is really the protein unbound drug that concentrates at the site of a target that is what leads to pharmacologically relevant activity. So primarily for many agents, once they're consumed or introduced into the system, they're protein bound. And through a complex set of interactions, some of which I've identified for you here on the right, there's a small fraction of that asset or agent that's going to be free. And that free drug is really what matters in terms of what is arriving in the brain tissue and what is permeating around the tumor cells themselves. Predicting free drug in human patients using preclinical data is something that's been fraught with peril. Here's an example of a WEE1 inhibitor that was examined in a pretty robust preclinical animal model using multi-mass spec to introduce a PDX. I've highlighted for you here where that tumor is, and that stippled green is the proportion of drug that's permeated the area. Overall, the analysis here in the preclinical data was that there was essentially no free drug permeating that tumor and very little total drug. These ratios called the KP and KPUU allude to that. The same drug, however, in the same tumor, but in actual patients, yielded completely different results. And these types of examples come over and over again. It's not to demonize the veracity of a preclinical model, but just to highlight that there are species-specific differences in the blood-brain barrier and in drug performance that just can't be correlated across these models. In the distant horizon, and now analyzing the performance of a drug probably won't require any tissue at all and could be done non-invasively with imaging. I don't think that our technology is quite there yet, but there have been efforts at this. AZD1390 is an ATM inhibitor that's just now entering randomized phase 2/3 study, but was designed from the ground up by AstraZeneca in order to be brain penetrant as an ATM inhibitor, and was basically derived from a backbone molecule that was an ATM inhibitor with systemic effect, but no CNS penetrance. And then the team at the company very intelligently redesigned the molecule in order to be this first-in-class brain penetrant selective ATM inhibitor that it is today. Now, when they tried to anticipate exactly how brain penetrant this would be, they used a pretty complicated set of models that relied upon radio-labeled carbon-11, 1390 in patients measured, and then inserted into sort of an in silico two tissue compartment model to predict the KPUU. When this same molecule then was entered into a phase 0 trial, which is something that our team did several years ago, it was in fact brain penetrant, but at a completely different level than what had been predicted. Just again, underscoring the fact that it's very difficult to predict the performance of these agents without asking the question in the patients themselves. And so this is where there's been this recent push to highlight the need for what are called PK/PD-driven clinical trials, which are effectively studies that humanize the animal model by asking PK/PD questions in the patients themselves. PK, of course, being the performance of the drug in terms of its CNS penetrance, and PD, of course, being the drug's end effects on its molecular targets. There was a recent review led by Mike Vogelbaum that nicely identified the scope of these trials and also the paucity of them over the last couple decades, underscoring the need to really elevate the importance of these studies, and from my perspective, underscoring the need for neurosurgeons to take a lead in proliferating these kinds of studies. We know that there's a range of clinical trial phases. Some of us are familiar with what are called microdosing phase 0s, where we're using literally 1/100th of a dose of a drug, which doesn't really have much applicability within a blood-brain barrier model. And then we are, of course, familiar with phase 1, 2, 3s. In between them, however, are these tissue-based trials, these PK/PD-driven trials, one of which is called a phase 0. A phase 0 trial is when a drug is introduced to the patient without any therapeutic intent prior to a tissue acquisition. That tissue acquisition occurs, and tumor PK and PD is queried. A phase 0 trial by itself is a non-therapeutic trial. There's no intended therapeutic benefit for the patient. A window of opportunity trial is when that drug has been introduced to the patient with therapeutic intent. So whereas in a phase 0, perhaps the patient will get 1 dose of drug, 2 doses of drug, maybe in a couple hours before the tissue acquisition, a couple days. In a window trial, that patient is likely getting drug for days or weeks or even months at a time because you're trying to treat something with the drug. And then there's this opportunity in the operating room or in the tissue acquisition window to acquire tissue, query PK/PD, and then the patient typically will go back on the drug at the same regimen with the same therapeutic intent. In between these two paradigms are what we call at the Ivy Center PK/PD triggered or hybrid phase 0 studies. And this study design is a little bit of a blend where drugs are introduced to the patient without therapeutic intent. So typically 1 day or a couple days of drug. And then the patient goes to the operating room and undergoes kind of immediate real-time PK/PD analysis of that drug's performance. If that PK/PD analysis demonstrates a positive response, the patient then continues on the drug, this time with therapeutic intent. And if there's a negative response or no evidence of drug effect, then that patient is off the study and is free to move on to any other experimental therapy. So this allows a little bit of precision in the deployment of the drug. And for patients, it allows some optionality, knowing that if they're going to stay on the drug or drugs long-term, that there's been some biological evidence of potential effect. And if they're not, then they don't need to spend any more time getting a very likely ineffective experimental therapy. It's important to note that all of these study paradigms can proceed or coincide or follow phase 1, 2, or 3. One of the most important monikers that we hear in the world of drug development for our patients in the brain tumor clinic is the concept of a brain penetrant drug. What's interesting to note is that there actually is today, there is no FDA or EMA definition of a brain penetrant drug. The regulators themselves will acknowledge that when a drug is presented to them, if the presenting team asserts that the drug is brain penetrant, they will largely accept whatever data set that team is providing in support of that statement. It could be anything from in silico data to preclinical data to clinical data, but there is no set standard. From our perspective, speaking from my perspective and our team at the Ivy Center, and I think many of us in the community, we believe that brain penetrance should be defined by the amount of unbound drug, meaning free drug, in gadolinium non-enhancing tumor tissue. Because as we all know, as neurosurgeons, resecting the enhancing tumor of a glioblastoma is very much the minimum of what we demand of ourselves in the operating room. It is that non-enhancing tumor that typically evades resection and is the basis of subsequent recurrences and therapeutic evasion. The operatic study was a window of opportunity trial using a early generation PARP inhibitor called olaparib, which was combined with temozolomide in recurrent GBM, where patients were exposed to the drug, then had tissue acquisition, and had some analysis of the concentration of the drug. And that analysis I'm sharing with you here. They tried to sample different compartments of the tumor based on tumor fluorescence. And as we know, tumor fluorescence doesn't entirely align with gadolinium and gadolinium non-enhancing compartments. Nevertheless, what they concluded was that there were comparatively high levels of total drug, and that was all they were measuring for this agent in these compartments. And that when they looked at what they called core versus margin regions of tumor, again, using tumor fluorescence, they saw some differential spread of this. Ultimately, they analyzed that there was approximately 100 to 1,000 nanomolars of drug, total drug, in these recurrent GBM patients, which was quite a high level, and which led them to conclude that this is a brain penetrant molecule and certainly served as a foundational basis for entering into a later phase randomized study. However, the same molecule was part of a phase 0-2 or a hybrid phase 0 analysis that our team performed using it versus another PARP inhibitor called Pharmacodynamic. And I won't belabor the study design, only to identify that one of the arms in the study used olaparib. And so we had the opportunity to really interrogate unbound concentrations of this same drug in gadolinium non-enhancing tumor compartments in an effort to identify patients that would be potentially benefited from subsequent therapy. What you see here in the vertical colorized bars is the gadolinium non-enhancing compartment of the tumor and its unbound concentration of olaparib. What you see in the horizontal dotted line is a minimum threshold of drug concentration that one would require to expect any sort of efficacy from olaparib. And so you see very easily that even in the first 5 patients, only one of them marginally exceeded this threshold, and the majority of them were well below it. And in fact, in total, we only measured 4 to 7 nanomolars of the same drug in its unbound state in the same tumor population. And so here are 2 different studies with the same agent leading to 2 completely oppositional conclusions in terms of the potential of an agent. The first study concluding that this agent should be accelerated into a late phase trial, and then a second study using PK/PD parameters, effectively identifying this agent as something that should not be developed at all for recurrent GBM patients. So what I will use this for is a basis to kind of convey to you that when you have real tumor PK data on any agent, it contextualizes the clinical data in a way that we can't really understand without knowing that PK data. So for example, this is another study, a really well done study with an FGFR inhibitor targeting FGFR aberrations in recurrent high-grade gliomas. What you see on the left is a waterfall plot demonstrating some subpopulation of patients, the ones that are dripping down, that had, at least in the study, some decline in tumor volume from baseline after exposure to the drug. And what you see on the right is the swim lanes for these same populations of patients with a subset that clearly had a better outcome than the majority. And so this is the kind of heterogeneous data that we see all the time in uncontrolled early phase studies. And it represents the biological heterogeneity of the disease. And it's difficult to interpret, but the logical interpretation of this data without anything else is that there are some subset of patients that may be responding to this agent, and therefore our analyses should then focus on identifying what is unique about this patient population in order to exploit them with this FGFR inhibitor. And that's exactly what the authors concluded. However, if you take the same agent and you enter it into a hybrid phase 0 study, as we did, you see the first 3 patients in that study and their colorized bars showing the unbound concentration of the same drug in the same patient population, and the horizontal dotted line being what you would like to achieve or more. You see that patient 1 exceeded that threshold in its drug concentration. Patient 2 barely met it. Patient 3 was well below it. So number 1, there's a degree of interpatient heterogeneity that we have to acknowledge when it comes to drug penetration. Number 2, in the top panel, what you see are the pharmacodynamic effects for these same 3 patients. Patient 1 has a very favorable pharmacodynamic effect. Patient 2, not so much. Patient 3, not at all. So the PK data gives you some predictivity of what potentially the PD effect will be. Now, 2 of these patients, based on the study criteria, continued to get the drug longitudinally for a longer period of time. You see patient 1 and patient 2, those horizontal lines on the bottom panel, neither of whom stayed on drug very long because of progression. However, one of them had progression and a reoperation, and we see that in fact the FGFR target we were targeting, which is a TAC3 fusion, was lost. So this gives you some understanding of why drugs can fail for these kinds of patient populations as well. And you wouldn't know that unless you ask that question. So tumor PK data really contextualizes or clarifies why we see heterogeneity from patient to patient in, for example, targeted inhibitor studies. What the data also tells us is that if we're just looking at tumor concentrations in gadolinium-enhancing tumor, it's going to vastly exaggerate how the drug is performing because, of course, gadolinium-enhancing tumor has a more permeable blood-brain barrier. And it also instructs us that CSF is not a very reliable correlate for a CNS penetration profile. At our program at the Ivy Center, we've done hundreds of patients on phase 0 trials, and we have yet to see any clear correlate between the CSF concentration of any drug across any drug class and the gadolinium non-enhancing unbound drug. So these are simply two different compartments within the brain, and one should not be used as a surrogate for the other. As neurosurgeons, I think we identify the importance of tissue in these kinds of studies, and specifically the importance of having a good comparator when you're trying to ask a pharmacodynamic question. So this is another clinical study that we recently reported. This is a CDK4/6 inhibitor plus an ERK inhibitor. And I won't belabor the logic for the biological pairing, but I'll just overview the study design itself, which is for patients with recurrent disease, recurrent high-grade gliomas, recurrent GBM, who have one of 3 molecular criteria that have to be met in order to enter the study. And so these patients have been previously resected, had Stoop regimen, qualified for the study, entered the phase 0 component, had several days of both drugs together, and these several days were then capped with an operation that occurred either 3 to 5 or 7 to 9 hours after the final drug dose. And that operation provided an opportunity for tissue acquisition from multiple compartments and for us to measure the unbound drug concentration of each drug, because it's a dual drug cocktail, in gadolinium non-enhancing tumor compared to this pharmacokinetic threshold, which we're defining as 5-fold of the biochemical IC50. And only patients that demonstrate both drugs in excess of this threshold then go on to a formal phase 2, which is statistically designed, as many are, as a 2-stage study. So when we're undertaking a study like this, the key question at the PD level is, what are you going to use as a readout for drug effect? And when it comes to an ERK inhibitor, there is no consensus on that. There are many logical or arguable candidates as biomarkers, and ultimately we settled on phospho-RSK. Now, to cut to the clinical readout for the study, it was unremarkable, and we really did not see any clinical signal from this pairing, and the question is, why is that? There is a sound logic for CDK4/6 and ERK inhibitor combination and for the synergy of that kind of pathway shutdown. When we look at the drugs themselves, what we see is that if we look across all of the tissue pairings, meaning their initial diagnosis tissue compared to the phase 0 tissue after being exposed to these drugs, we can use phospho-RB as a readout for CDK4/6 inhibition. We can use phospho-RSK as a readout for ERK, and we can look at a broader biomarker like MIB1, which is, of course, a proliferative index. And we see some trends here that may tell a story. But we have to recognize that it is about, again, the tissue comparison. And we all know that in glioblastoma, there's plenty of genetic drift that happens between initial diagnosis and recurrence, and plenty of variability introduced by the therapies that occur in that interval. And so this kind of comparison is not actually very productive because of that kind of genetic variability. What is productive is to use a pre-treatment biopsy, which we have the opportunity to do in a select number of patients where there's a needle biopsy occurring immediately before that exposure to the experimental therapy. And here the story is quite clear, which is that a CDK4/6 inhibitor like abemaciclib, which we know is brain penetrant because it's already been the subject of a phase 0 study by our group and others, demonstrating exceptional brain penetrance, is in fact shutting down what we would expect to be the biological readout for CDK4/6, which is phospho-Rb. However, if you look on the panel on the right, it's the ERK inhibitor that really fails to accomplish what it needs to do within the same population. Now, what I'm showing you are very limited snapshots of drug effect, and I'm making the point that having real proximal samples to drug exposure will give you clarity, which is, I think, self-evident. But serial needle biopsies itself is a rather tall order for the patient and for the healthcare system. And so we've moved on to what are called intracavitary Ommaya reservoirs for these phase 0 trial patients, where, as I'm showing here, an intracavitary Ommaya is placed at the time of the operation, and this allows us to detect tumor-derived DNA, RNA, cells as well, with a high degree of certainty and safety. And here's a case example of one such patient who entered a PARP inhibitor study, had that intracavitary Ommaya, and then around week 14 ended up demonstrating some evidence, or sorry, week 10, some evidence of emergence of a new variety of tumor signature, in this case in the form of an EGFR amplification that didn't previously exist. So this, of course, gave us some indication that there was a tumor progression event happening that had emerged de novo. And of course, several weeks later, the radiographic imaging caught up with that data and demonstrated this recurrence, ultimately leading to a second resection that was paired with a different clinical trial. Now, we knew that this patient had an EGFR amplification cooking, and so this allowed us to prepare that patient to be pivoted from one experimental therapy, which is the PARP inhibitor, to another, which is this brain-penetrant EGFR inhibitor from Black Diamond called BDTX-1535. And some of this data we recently reported at EANO 2024. So this kind of dynamic dosing of experimental therapies is enabled by horizontal surveillance of tumor genetics with approaches like CSF liquid biopsy. Now, I'll sort of zoom out here and try to put all of this strategy in the context of what we originally talked about, which is how do we get to late phase trials in a smarter and faster way in order to accelerate innovation in brain tumor patients and the opportunity for real positive signals in merited phase 3 studies. So I've mentioned a PARP inhibitor before. PARP inhibitors as a drug class are a recently emerging drug class that have had some success outside of neuro-oncology, specifically in the setting of other systemic cancers like ovarian and fallopian, really exploiting what's called a synthetic lethality mechanism, where these cancers have BRCA mutations that then, when exposed to PARP inhibition, lead to a synthetically lethal event in tumor cells. Now, brain tumor patients rarely have BRCA mutations, but it's been known that PARP inhibitors also modulate what's called DNA damage response. And so what you see on the left is a caricature demonstrating the two ways in which a PARP inhibitor effectively entangles the PARP molecule to damage DNA and prevents tumor cells from repairing their own damaged DNA, which occurs all the time, and therefore leads them into a cell death event. Now, it was originally thought that it was the direct catalytic inhibition of the PARP target by the molecule, which is what really mitigated this kind of effect. What we now understand is that it's a combination of effects between the direct catalytic inhibition and what's called allosteric trapping, which is a conformational event that decreases the ability of the PARP molecule to turn over. And different molecules in the drug class have different capabilities of each of these types of effects. So you see the spread on the right of different assets within the PARP class, pamiprib, veliparib, olaparib, etc. And what you see with the vertical y-axis is their ability to retain PARP at chromatin, which is equivalent to their ability to really potentiate DNA damage response, or rather to inhibit DNA damage response and potentiate something like, for example, a DNA damaging target, like a targeting strategy like alkylating agents like temozolomide or radiotherapy. Now, pamiparib, olaparib, we've already talked about a little bit, and they're definitely in the lower end of the spectrum. Veliparib, many of us are familiar with because it was the subject of a randomized phase 2 study several years ago that was completed by Mustafa Khosrow that demonstrated no benefit when you combine it with radiotherapy and temozolomide. We're also familiar with veliparib because it was the subject of a larger randomized phase 2/3 that was led by the Alliance that was also read out as negative not too long ago, where they combined the agent with temozolomide in sort of a chemosensitizing approach. Now, in retrospect, when we look at the entire picture, it perhaps is not surprising that these agents were less effective because of their limited ability for chromatin retention potentiation. On the other end of the spectrum is a molecule like Niraparib, which is certainly best in class in its ability to retain PARP damaged chromatin. Now, one of the ways in which we should be pursuing the selection of agents in a drug class when you have multiple opportunities is not to predetermine success, but to look at each of the drugs in the class and measure their performance head to head and try to understand which one has the better potential for our patient population. And that's exactly what we did with a combination of phase 0 trials that ultimately demonstrated that it was Niraparib that had the superior CNS penetration by far and superior pharmacodynamic modulation when you look at radiosensitization than all of the drugs in this class. We then selected this molecule, Niraparib, to enter a hybrid phase 0 study, combining it with radiation in newly diagnosed MGMT unmethylated patients. So these are patients that are freshly diagnosed, are unmethylated, and therefore will derive no statistical benefit from temozolomide. So you exclude temozolomide and instead dose them with Niraparib and give them conventional radiation as well, and then follow on with Niraparib monotherapy. Here we have the PK performance of the drug in these patients, which is best in class. And importantly, the free drug ratios, the KPUU on the right at 0.82 in non-enhancing tumor is very good. Here you have the pharmacodynamic effects of this combination, which again, on the scatter plot on the right, demonstrates the appropriate suppression of the pharmacodynamic readout for this agent, which is PAR, and the consequent radiosensitization when you're radiating these tissues ex vivo after being exposed to this drug in the patient. And here are the swim lanes at an initial readout that was reported at ASCO for the patients in this, albeit small scale, uncontrolled phase 2 component of the phase 0 study, demonstrating a median OS of 20 months and an OS 12 of 64%, with the median duration of treatment of 3.4 months. So a small sample size data set, of course, an unrandomized data set, of course, but something that's contextualized by strong and compelling PK/PD data. And so it's the balance of these data that then give the owner of the asset, which in this case is GSK, the confidence to make a decision to enter the CNS oncology space with this asset and pursue a large-scale global randomized phase 3 study, which is now what's underway, called the GLIOFocus trial. And this GLIOFocus trial randomizes newly diagnosed patients, just like the hybrid phase 0, to standard of care versus this Niraparib plus RT followed by Niraparib monotherapy. These are unmethylated patients, of course. It's a 450 patient study. It's a 1-to-1 randomization. It's a head-to-head comparison. So this entire sequence took less than 3 years to go from the concept of the phase 0 to the first patient in on the phase 3. So you have a very, very accentuated abbreviation of this life cycle of the decision for a drug, and you have a drug entering phase 3 with not only some clinical signal behind it, but also, importantly, biological signal at the PK/PD level. So if I can zoom out for all of us in our field, the points I would make are, one, preclinical data is very important when we're trying to validate new concepts in therapies for brain tumor patients, but it really needs to be cross-compared to human tissue data before we get ahead of ourselves. Two, the PK/PD readouts really need to be embedded into every early phase study so that you're contextualizing your results and making sure that there's not some other confounding element that's giving us a result that actually won't be borne out in the phase 3. And finally, this kind of strategy is how we start to accelerate our partnerships with the industry that is going to be the source of new drugs in the future, as it has been in the past. New drugs don't come from academic medical centers and medical schools. They come from biotech, biopharma. And so those industries have to be brought into the fold in ways that make sense for them in trying to clarify which assets merit their investment, and importantly, which assets need to be deprioritized, which is also a value add for them. What I've shown you a lot of is a product of the multidisciplinary Ivy Center team here in Phoenix at the Barrow Neurological Institute. This is a team that has been built from the ground up simply to pursue early phase PK/PD studies and usher new drugs from the late preclinical to the early clinical and now into the late phase clinical phases in support of brain cancer patients. And so I'll stop there, and I'll thank you for your time and attention.
Thank you, Dr. Sanai. That was an amazing talk. I think highlighting a lot of challenges and opportunities, and I think also highlighting the neurosurgeon's role in guiding therapies, which is something that I've always felt very strongly with in terms of not just operating on patients, but developing strategies to move on to larger randomized studies that are meaningful. So we're going to have a couple questions, and I know Maria is going to have some questions. Nader, one of the things I know you're working on and others to enhance the blood-brain barrier disruption is focused ultrasound. And you showed us a nice study here at the end with a drug that does get into the non-enhancing tumor. Do you think focused ultrasound can lower the bar for us and help us with drugs that work well in other cancers, but may work better in the brain with focused ultrasound?
Yeah, thanks, Costas. I think it's a really important space for us to pay attention to. The early data in focused ultrasound is promising. A lot of the early studies, preclinical and clinical, are demonstrating that there is some disruption of the blood-brain barrier. And the way they read that out, of course, is by the presence or absence of, or increase or decrease of gadolinium. And so what those studies tell us is that for the molecule gadolinium, which is a very small molecule, there's a window of time after exposure and focused ultrasound where it's more permeable and you're getting an increase in permeability of the blood-brain barrier to that molecule. The next step now is to really ask and answer that same question on a drug class by drug class basis, because of course, every drug is going to have a different set of kind of biophysical properties than gadolinium. And it's really within the context of those individual molecules that we need to ask and answer the question. It may be different from class to class. One of the key questions for any sort of mechanism of disrupting the blood-brain barrier, whether it's focused ultrasound or a chemical disruption, et cetera, is what is the scale and scope of that disruption, as well as what is the temporal window of that disruption? And then how do you pair those things with the actual administration of an experimental therapy? So I think I'm personally very optimistic about the role of targeted ultrasound. There are some technical obstacles to overcome in terms of its ability to disrupt large territories of the brain, which is what you would want for any adjuvant therapy strategy. And then there are some more specific questions about, well, what is the performance of a drug with or without this strategy when we're looking at newer drug classes, the things that are currently in development for other oncological indications, where the PK profile is not quite so clear.
Sir, I believe that there are also the nanoparticle related drug delivery systems that are being considered. And also, sir, you have a very wonderful work on the super selective intraarterial drug delivery system, sir. So I guess that these are all the other systems that we might use in the future to have some edge in drug delivery.
Yeah, that's right, Maria. So one of the realities of our space is that we spend, as a collective community, neurosurgeons and CNS oncology in general, an inordinate amount of effort and resources on the discovery side of brain tumor biology, as well as the targeting side and drug development side. But there's comparatively little effort and resource allocated to drug delivery itself. Nanoparticles, intraarterial disruption with hyperosmolar agents, targeted ultrasound, these are all important examples of opportunities, but still comparatively limited in terms of the number of centers and teams pursuing them. So I think it tells us that strategically we should be shifting more and more efforts to the delivery side of things, and try to be a little bit more rigorous in terms of how deeply we're examining these strategies and how we're trying to pair them with therapeutic innovations. But you're absolutely right, Maria. Those are all important frontiers. Many of them at our program and others are in investigation.
Thank you very much, sir. Sir, there's also some news like studies like photodynamic therapy and exploiting of necroptosis. Sir, do you think that this could be something that might lead us somewhere better for those tumors, especially those glioblastomas which are drug resistant? Do you think that do you see them as good strategies?
Yeah, absolutely. I mean, I think we have Kostas here who could speak to photodynamic therapy and the importance of leveraging the opportunities that 5-ALA provides better than anyone. Photodynamic therapy and its kind of pairing, which is sonodynamic therapy, are all, in my mind, metabolic targeting strategies where you're exploiting metabolic inefficiencies in the tumor in either invasive or non-invasive ways. In these strategies, exploiting the accumulation of protoporphyrin IX and the sort of lethal events that may be happening when you expose that protoporphyrin IX to some sort of subcellular excitation. I think that these are really, really innovative approaches. Again, approaches that really merit wider attention within our community, and approaches that will never be solved without a surgically driven trial approach. And in all of those examples you provided, we have neurosurgeons that are leading a lot of the architecture of the experimental therapy designs. And I think this is an inspiration for our community. Drug development and therapeutic development has historically been outside of the purvey of neurosurgeons for many years. But what we're learning now is that the lines are blurred, and a lot of the opportunities for answers are going to come from the operating room. And for many of us neurosurgeons, we're as involved in the adjuvant therapy of these patients that we treat as we are in the operation.
Thank you very much. So there are also some studies like which are discussing the neoadjuvant immunotherapy for tumors, and there is also some sort of a concept where we can give a drug cocktail to patients where they actually can reprogram the glioblastoma cells into something which is more likely resemble the native cells and which are no longer malignant. So they are like this concept exploits the fact that the tumor cells can be actually reprogrammed into something much more benign. Sir, do you think that this could be some other concept? I think this is a very wide discussion. If we just go on discussing about this, I think we are taking a much more time from you today, but it's just a question that just keeps on resonating inside my mind.
Yeah, it's a great concept, Maria, and it's certainly something early in its germinal stages. Neoadjuvant therapy has some precedent outside of CNS oncology. We've had some kind of index studies within brain tumors using neoadjuvant therapy, whether it's been in the setting of radiation, but more recently, I think you're alluding to checkpoint inhibitors. And then also this concept of, can we sort of limit the dedifferentiation of tumors as they go through their natural history? I mean, I think these are all important questions. I don't really have any answers to them. All I can say is that these are questions that can only be answered in the patients themselves, because our preclinical models are just inadequate to recapitulate the biology and the complexity of what we're facing in the clinic. And so there are some investigators working on some of these areas, certainly in the immunotherapy space, this concept of priming the tumor pre-surgically, then operating, and then exploiting it to a checkpoint inhibitor strategy is something that has a logical narrative behind it, and the studies will have to bear it out.
Well, we're running out of time, but I think this is an amazing talk that really highlights a career of work Dr. Sanai has really laid out for us to learn from. I think the PK/PD work, I just can't also stress that enough, and really seeing that guide drug development, again, led by neurosurgery, which is incredible. So congrats, Nader. You're doing some great work. Really happy with that. And thank you again for, spending Saturday morning with us, and everybody who joined. This is going to be recorded, so this is going to be available in perpetuity. So please view it at your leisure when you can. And I want to thank everyone again in the WNS and the tumor section, and Maria for another wonderful webinar. Thank you very much, everybody.
Thank you.
Thanks.